Some genes are linked to the X or Y chromosome, which means males and females are affected differently. Males are primarily affected by color blindness, where calico cats are typically female cats. We're going to learn about this today. So, scientists were able to identify the source of heredity as chromosomes. And our genes are carried on our chromosomes, and the chromosomes are the units of hereditary structure. So, chromosomes separate during meiosis, and each gamete then has half the number of chromosomes as a somatic cell. And our somatic cells are everything except the sex cells, the sperm, and the eggs. During meiosis, these chromosomes assort independently, so certain genes aren't tied to other genes during that process, and every chromosome can contain many genes. And all of our chromosomes are autosomes. We have 22 pairs of them, except pairs 23, which is our X and Y sex chromosomes. So Thomas Hunt Morgan, an American geneticist who used fruit flies, and he used them to study Mendel's principles of inheritance. So he used fruit flies because they reproduce rapidly. They produce more than 100 eggs per mating soon after leaving the eggs. And so that's good for genetic probability because there's a lot of offspring. They typically have a very short life cycle of 10 to 15 days. So you can study many generations in a short period of time. Also, because they're small, you can house a lot of them. And males are easily distinguishable from females. So Morgan discovered that different mutations were linked to certain traits. So this supported the theory that genes responsible for traits are located on the same chromosome if they're linked together. He found that he had one white-eyed male among many red-eyed offspring, and so he concluded that white eyes must be a mutation. He mated that white-eye male with a red-eye female, and all of the first-generation offspring had red eyes, which would be dominant. But when he crossed two F1 hybrids for an F2 generation, he got 3/4 red and 1/4 white. This was explained by Mendel, but Morgan also found that only males had white eyes, no females. After further crosses, he found that females could express the white mutation, but not in the F1 or F2 generation. To explain this, Morgan decided to study cells from the salivary glands of Drosophila, and they only have about 8 chromosomes. He stained these chromosomes and viewed them under a microscope. He found that males only had 3 homologous chromosome pairs. The sex chromosomes weren't identical, and females had 4. So therefore, our females would be XX and males were XY. Therefore, he made the conclusion that because X and Y are not completely homologous, they must contain different genes, unlike our autosomes. Drosophila eye color is located on the X chromosome, but because Y is not identical, there is no eye color gene located on the Y chromosome. And this led to the idea that there are sex-linked traits, or traits that are controlled by genes on the sex chromosomes. Therefore, a red-eyed female could be red-red, dominant, heterozygous dominant, or homozygous dominant or heterozygous, but a white-eyed female would have to be homozygous recessive. A white-eyed male could be have a dominant recessive gene, or a red-eyed male would have a one dominant gene because the red gene was located on the X chromosome. So here's our explanation of the F1 and F2 generation. So if you had a red-eyed female and a white-eyed male, because all of the female's X chromosomes had a red-eyed gene, none of the offspring, male or female, could have white eyes. However, the two female offspring will carry the recessive allele. So if you cross 2 of those F1 offspring, one male and one female, one of the male offspring could carry the white-eyed gene on their only X chromosome. So male offspring will always inherit an X-linked trait from their mother because the father would supply the Y chromosome every single time. Females can have 3 possible genotypes. They could be heterozygous, homozygous dominant, or homozygous recessive. And males can only have 2. They cannot be heterozygous for a sex-linked trait because they only have one X chromosome. So that means they either have it or they don't. To get a white-eyed female, a female with at least one white gene, so heterozygous, or homozygous recessive, must be crossed with a white-eyed male. So sex-linked genes are also found in humans. For example, red-green color blindness, which is determined by a recessive trait on the X chromosome. Males are colorblind much more than females because females need 2 copies of the recessive gene, one on each X chromosome, to be colorblind, where the males only need 1. Roughly 10% of males experience color blindness compared to only 0.4% of females. So this is a standard color blindness test you might see at a ophthalmologist office, and sometimes they have patterns in them where people who are colorblind can see numbers, but people who have normal color vision cannot see numbers. So let's do a few examples. So the color blindness is a sex-linked recessive disease. So in our Punnett square, we have to put our X and Y chromosomes. So an affected male marries a heterozygous female. So we're going to go, she is heterozygous, so we're going to go X normal, X colorblind. And then an affected male will be X colorblind, Y. So that's what we're going to put in our Punnett square. And we want to put those sex chromosomes so we know if we're having male or female offspring and which ones are going to be affected. So you definitely have to put those X's, not just colorblind, yes or no. So when we go through the Punnett square, what is the chance that they will have an affected child? So when we look, everything in this column could be affected. So it would be a 50% chance that they have an affected child. Could their daughters be affected? Yes, their daughters can be affected because the male is affected. So all of his daughters will get at least one color blindness chromosome. And because the female is a carrier, yes, their daughter can be affected. She can have both recessive genes. A second example, suppose hairy ears is a Y-linked trait. A man with hairy ears marries a woman with normal ears. So we can look at, oh, it's a Y-linked trait, so I have to erase that. So there's nothing on the X chromosome. She would be X. X are her two options. This would be X and then hairy ears. So we can put these in. So when we fill in our Punnett square, their daughters are not affected, but both sons will be. So could any of their children have hairy ears? We can say yes, but only the sons could have hairy ears. The daughters cannot because it is not a gene that is carried on the Y chromosome. So therefore female, or the X chromosome, therefore females cannot be affected. Okay. Now we're going to look at a disease that's inherited as a sex-linked dominant trait. So this an affected male marries a homozygous recessive female. Homozygous recessive female, let's just use the letter H. So she is homozygous recessive for this trait. So she is going to have two recessive alleles, and a affected man marries a recessive allele. So remember, this is a dominant trait. So he has this disorder. So when we look at the females' chromosomes, then the male's chromosomes. So when we look at their children, is what's the likelihood of any of the children being affected? We're going to say yes, daughters at 100%, because every X chromosome they get from their father will have the dominant trait, and that means they will have that disease. Would the parents of the father also have been affected? We'll say yes. The mother, because she's the one who passed on that affected X chromosome to her son. The likelihood of their male child being affected, none. And the female child being affected, 100%. So we can do any of these Punnett squares for sex-linked traits, but again, it's really important to identify the chromosomes, the X and the Y chromosome, and then the trait as a superscript. So bar bodies are small dark spots of chromatin that are located in the nuclei of female cells, and they're not found in male cells. Um, in 1961, Mary Lyon proposed that the dark spot was a dormant X chromosome because one of the X chromosomes will become inactive, but this inactive chromosome varies between cells. So not all female cells are identical depending on which of the two X chromosome is active. And heterozygous females who carry a lethal gene on their X chromosome may escape disease because this gene is only active in about 50% of their cells. So if we look at this image at XY cells have two distinct kind of nuclei, where females have one and then a small dark body on the edge of the cell. So we can see this in calico cats because male cats typically are either going to be black or orange because the gene for color is carried on the X chromosome and they only have one, where females can be black by having two black chromosome alleles, orange by having two orange alleles, or they can be calico by having one black and one orange allele. So there's areas on their skin where the orange allele is active and will have orange hair, and areas on their skin where the black allele is active and they'll have black hair. And male calico cats cannot be cats can't be calico unless non-disjunction occurs and they have an extra X chromosome. So they would be XXY. And these cats would then be infertile. So male calico cats are very unlikely, but if you do find one, they won't be able to have any offspring. Looking at some other sex differences, TDF is testy determining factor. And humans have 46 chromosomes. Females have 23 pairs of homologous, 22 autosomes, and 2 X chromosomes, where males have 22 homologous chromosomes and then 1 X and 1 Y. So they don't pair up equally. The human X chromosome carries 100 to 200 different genetic traits, where the Y chromosome only codes for gender, and this is called testes determining factor. Now, this isn't active until roughly the 6th or 7th week of pregnancy, and therefore, until that point, a male fetus is no different than a female fetus. The testes develop in the body like ovaries, but until testes determining factor is activated, then they would descend and begin to produce testosterone. When we look at testicular feminization syndrome, these are individuals that appear female even though they have an X chromosome in their karyotype. This syndrome is caused by a mutation on the X chromosome that acts only in the XY zygotes because there's only one X chromosome. They don't react to injections of male sex hormones, therefore they don't display the muscle development of males. So they don't respond, their bodies don't respond to testosterone in the body cells, so they continue to appear female. Fragile X syndrome is another condition that primarily affects males because they have only one X chromosome. And the symptoms of mental disability range depending on whether the person has a more severe case and they're male and they only have one X chromosome, or if they're female, which is less severe because they have one normal X chromosome and one atypical X chromosome. And this can be inherited from either the father or the mother because sperm can have an X chromosome or a Y chromosome. So that's a little bit about the differences in sex and how it can affect our genes. I hope you enjoyed this lesson.